This is it.
Uh-oh.
Okay, when you put terminology, are you just talking about the basic terms?
They're in the
You're talking about
paragraphs of the lab manual exercise.
Ms. Peterson, you have to start breaking stuff down, okay? Your study
We
hall
have
is
like one more day left, and now you want to start breaking stuff down? Hey.
We're at the end
Okay,
here.
okay, Dana.
Huh?
That exercise was about
That was
pure cultures, right?
yes.
I'll
There
pull
were
it up.
two techniques. What were they?
Radiant Streak
Streak
and
Poor plate.
Four plate.
What was the first one?
Radiant streak.
Radiant streak and pour plate, okay. Why did we do those exercises?
to see
Isolated pure
Isolate.
culture.
To isolate what?
Pure culture.
Pure cultures. There's terminology. From
Oh,
a
okay.
mixture of species. What's a mixture? How would you define a mixture versus
More
a
than
pure
one.
culture?
Than one.
Okay. It's not necessarily complicated terminology.
Oh, okay.
Okay. But it's in the lab manual in those introductory paragraphs.
I don't have that manual.
Okay.
I ain't never had that manual.
I don't have one.
Then the next thing is, do you know the procedure for radiant streak plate?
Radiant streak was the drop and then
The drop
the two
and
lines.
then lines across.
And so how did that isolate pure cultures?
Because
By
it-
Because you only
decreasing.
did one
Yeah, it
drop, so you
decreased
dragged it
the
away.
So you were
amount
physically
per spread.
spreading the cells apart from each other, right?
Yeah.
Yeah.
And
Okay.
then one cell turns into one column.
Now, in the other procedure, the pour plate, how did we isolate pure cultures? We didn't use an inoculating loop, so
Test
that's
tube and we, yeah.
Yeah, we decreased the amount of the specimen.
the amount of what?
The specimen.
Okay, the number of cells decreased from one tube to the next, and
Yeah.
subsequently from one petri dish to the next.
Yeah.
Yes.
Mhm.
So what
Okay.
we did there was we isolated pure culture.
So what I'm asking you, I think you can figure out is, how did this technique isolate pure cultures from a mixture? How did this one do that?
By
Okay.
decreasing the number of
Okay.
cells.
Okay.
If you have questions, you gotta ask.
If you inoculate her.
Now, the last thing we did after we finished those procedures is we compared them.
Yes.
Which one was easier?
The
board plate.
radiant streak.
What do you mean by
Radiant streak.
easier?
Why was the radiant easier than the pour plate?
Because it had the colorful stuff.
What?
I don't know.
Because the colonies were different.
We had the
colors.
same bacteria in both techniques.
No, that was.
Oh, you talking about the?
Oh, because the port plate,
Oh.
you have to do more of the port, right?
They were aerobes, so they grew on top.
Wait.
What was the original
Listen
question?
to my question.
You said which one was easier?
Which technique was easier to perform?
I'd say the radiant streak,
I agree with you.
because there wasn't. as
What
much
made
stress.
the pour plate more complicated?
You had to keep it at a temperature, and you had to make sure you got it before it solidified. And
And you
Yeah,
had to
medium.
explain your oils go in between.
And you
Yeah.
had to pour while it was still liquid and
Yeah.
all,
And that
so
was a timing
it was very
issue on it.
time
So-
sensitive.
Yeah.
Okay?
Mhm.
Now, when we compared them in terms of results, Dana,
Okay.
what kinds of microbes could we grow with the pour plate technique?
The pour plate can grow any of them. The
All right, what are the
aerobes-
any?
Aerobes and anaerobes and facilitative.
Facultative. Facultative.
All semester you've been in here,
I
you still haven't
know.
learned that word.
I still have it.
Okay?
That's
And
all.
what about the radiant streak method? What kinds could we grow there?
Aerobes.
And
Facilitative.
Facultative.
Facultative.
what could we not grow with the streak plate technique?
Anaerobes.
Okay.
Yeah.
Because I gotta be in the
They need
Medium.
oxygen. I mean, they
No.
don't
They
need
need
oxygen.
no
They
oxygen.
don't need
So
oxygen.
where would the anaerobes grow in the pour plate technique?
at the underneath the medium.
In the medium or under the surface, however you want to say that? And where would the aerobes be growing?
Surface only.
Surface.
And where would the facultative ones grow?
Possibly
Everywhere.
anywhere.
In both locations, right?
Every pour plate takes longer.
Now,
Yeah.
there was also a handout that you had for that week's lab work that's entitled Special Microbiological Media.
Oh, yeah.
Oh, yeah.
I'm was missing one. Is that the one?
See, because mine just says microbiological media. Maybe it is. Oh.
It is micromedia, and then the two subcategories were
Oh, special and selective.
There we go.
No, special.
nutritive and special, right?
I've got, yes, good. Nutritive
And
and
then
special.
under the special category, there were three subtypes.
Enriched,
Selective,
selective, and different.
then I forgot which came next.
It was
Differential and
enrichment,
No, I got that one.
enriched,
selective,
selective, and differential,
differential.
in that order. Okay. Now, do you know what makes a medium enriched versus differential versus selective?
Enrich, something's added into it.
Yeah, so is there for the differential?
Well, it's added
It's
to
for
promote growth.
fastidious growth?
So it's a growth factor?
Yes.
Yes.
Okay. And in differential?
That one is, it
It's-
inhibits everything.
it's an
Oh.
inhibitory thing.
That's selective.
Oh.
And differential?
Okay,
Oh.
Amy, and everybody else, my point is, you have to know the definitions for those three kinds of media.
Okay.
Now, you actually had to know that on a lecture test already,
Did we?
I probably missed
so
that.
that should not be new information.
Mr. Peterson, you put so much information in my brain.
I agree.
I'm so grateful you tell me that, Dana,
I
because it
don't know
means
if-
I did what I was supposed to do.
But I don't know if I have the ability to
Like 10?
go into the catalog and find it because
That file cabinet is stuffed.
it's
full.
stuffed and
Yeah.
there is no sort of filing system. It was like, go.
That was my problem on the last episode. I kept saying, what was he talking? What did he say that day? And I'm like,
I just feel like
God, there
there's
was
so many
much
weeks.
we were talking
I
about
have
that.
no doubt that you have that capability.
You have so much
That's
faith
very
in
nice
us.
of
I
you
don't
to say.
understand.
Much.
And I mean that for every student in the class,
Know the difference.
okay?
Define.
All right,
Okay.
next.
Now, I have a question. Do we just need to know the enrichment, selective, and differential, or do we have to know the different types of each of those too? Like because some of them I
Like the
don't
blood
remember us using,
auger.
like the brilliant green, the transbro auger, staphylococcus medium, the EMB auger.
What's in staff medium that makes it
Well,
select?
staff. So we do need to know those, but
Yes.
just the basic, what's in them. Okay.
So we
Do tell me how you would
like,
formulate a
What is that?
question
Right here.
Okay.
on Brilliant
So
Green Auger.
You need to know what's in brilliant green auger. You need to know what makes it
But it's really
brilliant
green
green.
dye.
Which of these media
That's the question.
is used to isolate salmonella from a mixture of
Brilliant
enteric
green.
Green.
bacteria?
That's how the questions will be. That's what I'm saying. I just need to understand how you.
So, in answer to your question, Amy, yes, you have to know about those
But
kinds
just
of
the
media.
basics though. Like salmonella, and then it's brilliant green dye is what isolates
Just like-
salmonella.
What makes it selective?
The salmonella.
No.
No.
What makes it selective
For
for
someone.
salmonella?
The green dye,
The dye.
because it
Okay.
inhibits most
What
enteric
makes
species.
staphylococcal medium selective?
The addition of staphylococcus.
Listen to my question. What makes the medium selective?
The concentration of
Salt.
sodium,
Salt.
Salt.
salt, NaCl.
What kinds of bacteria can grow in the presence of 7.5% salt?
Halophiles.
Halophiles.
Oh, okay.
So there were those were some specific examples, but what it should also tell you is this is how you have to study the other
Yeah,
information there,
okay,
Okay.
okay?
got it.
Let's see.
I think that's how I.
Exercise 2-8. This was the experiment where we inoculated media and then incubated the microbes at 5, 25, 36, and 50 degrees.
Oh, okay.
Okay? Was there some terminology you have to know there, Dana?
Yeah.
Yes.
Like what?
Let me get to that page.
Thermophile.
Thermophile.
Cycrophiles, mesophiles. And do you have to be able to associate certain temperatures with those terms?
Yes.
Okay.
5 Celsius is cyclophile, 25 to 36 is mesophile, and 50 is thermophile.
Okay.
How the
Stop looking.
hell do you have that in your brain?
Yeah, where are you seeing that?
So now on the review sheet, it says you have to know terminology. Dana's given us some examples of that. But it also says you've got to be able to interpret data. That means there's going to be a chart
Up there.
Oh boy.
with data on it. And you got to be able to interpret the chart. What would you have to figure out?
Is it a mesophile?
There you go.
Cyclophile or
Is this microbe X a mesophile, a thermophile, or a psychrophile?
But what's going to be on the chart? Like, what kind of examples would be on the chart?
Like it would show the temperature.
Oh, so it's going to have the temp and then.
Yeah, and if there's a plus.
And the amount of growth.
Okay, yeah.
Remember, you had to evaluate the amount of growth. Did it have 1 plus or 4 pluses? Okay?
Where
It's
is,
on the result sheet. That's the exercise.
why is
Now, the next exercise was not one from the lab manual. That's why in the margin it says handout. It says you're supposed to be able to interpret data resulting from the thermal death point determination experiment. Is there some terminology you have to know there, Dana?
Yes.
Like what?
Where we at? Oh. You have to know what thermal death
I was
point
gonna
is.
say,
There
have
you
to
go.
Know where the thermal.
You got to know what thermal death point means if you're going to interpret data that asks you what is the thermal death point for microbe X.
it's the temperature for
Oh,
which
okay, yeah,
at
yeah,
10
yeah.
minutes? Is that what it is?
Is it any temperature that killed them in 10 minutes?
It's the lowest
I think
temperature
it's the
that for
lowest.
10 minutes consecutively will kill a microbe.
There you go. So there is a specific definition. It's not just any temperature.
Lowest.
Okay, so there's terminology for you to know, and then there's interpretation of data.
And that was either 0 or plus, right?
I'll have Pedro, I will have the code on the test, what the symbols mean. Okay?
Where are my pictures?
Next is exercise 2-10, be able to interpret data resulting from the experiment, no terminology.
That was the salt.
This was the salt and sugar. All right?
Alright.
Some people put salt in the medium, some put sugar in the medium,
Mm-hmm.
and then we were trying to determine if the salt or sugar had an inhibitory effect or not,
Sugar it fed everything.
and the sugar did not have an inhibitory effect at the concentrations we used. In fact, it stimulated growth. But salt inhibited it, with the exception of what kind of microbes?
Halophilia.
Yes, sir. He was right. Halophiles. Okay?
I can't remember which ones that were. Which ones were?
You don't have to memorize that. You have to
Able to interpret the
use the data to determine
So
that.
it'd be like, oh, microbe X.
That's right.
Yeah. Okay.
data. Okay. Can you have a lot of those?
So it's not going to be like the names of microbes.
'Cause that's,
like it's
like,
on the data
relevant.
chart.
I could put names or I could put microbe X, Y, or Z.
But you just need to.
But you
look
don't
at
have
what
to
you have.
know that microbe Z is a halophile unless the data can show you that.
Okay. So, on your on this test, how many questions are going to be interpreting data, as opposed to?
I don't have that memorized. I couldn't tell you that.
Don't worry, Mr.
Could you
Eason.
tell
We'll
me
need
something
time to
now
look
and then just hold true to it? Say 45%, and then make sure that 45% of the test is interpreting data.
I would guess it'd be around that number.
Yeah, that's okay.
So out of 50 questions, approximately 15 to 20.
I'm guessing.
Just make sure when you formulating this test, when you're putting it together and you're doing it on your word.
He already put it together. Bruce,
I already have this test ready.
Yeah.
I don't have your final ready.
Go ahead and go across the hall and get it, Mr.
Your office is right there, sir.
Go ahead.
See, you're
We'll
just
wait.
from here.
We're not interrupting.
You know,